Hello and welcome to Teaching Bio. Today we're going to look at the control of blood water potential via the different mechanisms of the kidney. Now, this topic can be quite challenging to some people because of the many different mechanisms involved, and it can be quite confusing at times. However, I'm going to take each part of the kidney and the nephron and sort of look at the journey that the ions and the water molecules will take to eventually becoming reabsorbed and producing urine. So hopefully that should be done in an accessible fashion. Okay, so this is an overview of the structure of the kidney. Now, you don't need to know the structure of the kidney, however, it will help you sort of picture each of the different concepts better if you do have a brief understanding of what is going on. So we've got the renal artery, the renal vein, and the ureter. So the renal artery will obviously bring oxygenated blood, rich in lots of different ions, nutrients, and obviously water, towards the kidney. And it will effectively bring it into these parts of the kidney here. And this is the renal medulla. Okay, so that's indicated there, that region. This is the renal medulla. The bit outside is the renal cortex, but you don't need to be worried about each of the different names. Within the renal medulla, we have many nephrons, and it's the nephrons that are very important because of the very different mechanisms that each of the different parts of the nephron have to reabsorb certain ions and reabsorb water. Eventually, the nephrons will produce filtrate or urine, and each of the many nephrons in all of these different medullas, in these sort of pyramids, will feed all of these into this sort of calyx here, and that's part of the ureter. So they will feed into the minor calyx, they will join to form the major calyx, and then all of that will be drained into the ureter, which will then travel along to the bladder and to then be excreted as urine. So these are the different parts of the nephrons, those weird loop things that I showed you in the previous diagram in the medulla. This is what it looks like, and you do need to know the structure of the nephron. So you need to know that this first part is called Bowman's capsule, and we've got the proximal convoluted tubule. We've then got the descending limb, and this is the descending limb of the loop of Henle, okay? And then we go on to the ascending limb of the loop of Henle, and then we have a distal convoluted tubule. So there's proximal and there's a distal. Proximal just means sort of closer to the start, and distal just means further away. And then we move on to the collecting duct. And then this will obviously go on to the ureter and therefore go to the bladder. Okay, so the first step in the reabsorption of water, and obviously the production and the controlled water potential and the production of urine, is ultrafiltration, and this occurs at Bowman's capsule. So this is Bowman's capsule, and this is the lumen of Bowman's capsule. Now, there's many different parts to Bowman's capsule, namely that there's this bundle of capillaries here, and this is called a glomerulus, and it has an afferent and an efferent end. So this is the glomerulus, and it's composed of this bundle of complex capillaries known as glomerular capillaries. So if we sort of take a look at the glomerular capillary in a more pictorial format, we can see the afferent arteriole and then the efferent arteriole. So blood flows from the afferent to the efferent arteriole. And this sort of space here would be the lumen of Bowman's capsule, as we looked at before. Now, between the squamous endothelial cells of the capillary, we have gaps, okay? These gaps are called fenestrations, and these gaps are super important to allow for small substances to leave, which is effectively what happens. So because of the high pressure, the afferent arteriole, because of the high pressure of blood, it will force water and lots of small molecules, such as small proteins and urea and glucose and sodium and chloride and calcium, out through the gaps in the squamous endothelial cells. So blood plasma enters Bowman's capsule via the afferent arteriole here. Blood travels through the arteriole into the glomerulus, which is the bundle of capillaries. Because the capillary is only one cell thick, there's this really, high hydrostatic pressure that's going to force all of the ions and solutes and small proteins and things that are big enough to fit through those gaps through the squamous endothelium into the lumen of Bowman's capsule, and that will then go on to form the glomerular filtrate. And they are glucose, amino acids, and urea, as I said before, and this mechanism is actually quite important in regulating blood pressure. So if we look at the afferent and efferent arterioles, this is the afferent arteriole, this is the efferent arteriole. Now what we can do is we can have different compounds in the body that can constrict or dilate the afferent or efferent arteriole. So if we constrict the afferent arteriole, and that's called vasoconstriction, so if we release some compounds to constrict that, well, what that will do is that will reduce blood flow into the glomerulus, which makes sense because obviously there's more resistance there because we're forcing this pipe. So we will have less blood moving through into the glomerulus. Therefore, we have a decrease in the glomerular hydrostatic pressure and then a decrease in the glomerular filtration rate, meaning that we will have less formation of filtrate. Vice versa, if we use compounds to dilate the afferent arteriole, we will instead have more blood moving into the glomerulus. Therefore, we'll have an increase in glomerular hydrostatic pressure and then an increase in GFR. Now, the opposite is true for the efferent arteriole. So if we constrict the efferent arteriole, we will reduce the blood flow out of the glomerulus back into normal circulation. So because of that, we'll get this backing up of blood and this back pressure into the glomerulus because the blood's trying as hard as it can to force itself out the efferent arteriole, but it can't because there's too much resistance because we're squeezing into the pipe. So because of this back pressure, we'll have an increase in the GHP and therefore an increase in the GFR. And if we dilate the efferent arteriole, we get the opposite story because we're going to have lots of blood leaving. So we're going to have less of this back pressure. So we'll have less GHP and less GFR. So as we move along the Bowman's capsule, we move on to the proximal convoluted tubule. And here we get selective reabsorption. This is where we get the absorption of glucose, amino acids, water, and ions by active transport via the different mechanisms and sort of like secondary active transport, which we'll look at in a minute. These have different adaptations you need to be aware of, such as microvilli for a larger surface area. So there's a fast movement of ions and many mitochondria to provide ATP for the different sodium-potassium pumps to power this mechanism. So this is sort of a diagram showing the layout of the cellular structure at the proximal convoluted tubule. So this is the lumen of the PCT. Okay, so this is after Bowman's capsule. We've moved all that stuff out and we've got it here. And these are the epithelial cells that will be lining the surface of the PCT. Now, on the membrane epithelial cells, we've got a sodium-potassium ATPase. So that will use ATP to move sodium out into the blood and move potassium into the cells. And we've got this GLUT transporter, which is a glucose uptake transporter, which will move the glucose from the cell into the blood. So as I said, we've got sodium and we've got potassium. And that uses ATP to move sodium into the blood and potassium into the cell. That will decrease the concentration of sodium in the cell and increase the concentration of potassium. And that's very important that we have this pump because we need to have low levels of sodium in this cell here. So, as we've gone along, the urine's flowing, the filtrate's moving along, and with it, it's bringing glucose and sodium, which were freely filtered at Bowman's capsule. Now there's a transporter here, which is the sodium glucose cotransporter, and this is very important to bring sodium and glucose in. And what will happen is the sodium and glucose will couple together to move through the transporter into the epithelial cell. Now, I already told you before that the sodium-potassium pump moves sodium into the blood, lowering the concentration of sodium in the epithelial cell. As a result, sodium can move down the concentration gradient. There's always a higher concentration of glucose in the cell, obviously, because a cell will be actively respiring, so it will need a large, abundant store of glucose. So glucose will always be higher in the cell than in the lumen of the PCT. So glucose needs to move against its gradient. And the movement of sodium down its gradient is what provides glucose the energy to move against its gradient. Because as one moves down, it pulls the other against. So sodium moves into the cell and then uses this sodium-potassium ATPase to move back out into the blood, continuously lowering the concentration of sodium here so that more sodium can come in and with it bring glucose. And the glucose that is moved in will use our friend the GLUT transporter to move out into the blood and along, therefore being reabsorbed. Now the same story is true for amino acids. So it's the exact same mechanism, sodium in the blood, potassium in the cell, Filtrate moves along, amino acids and sodium. There's a sodium amino acid cotransporter. Sodium will move down its gradient, and that will then provide energy for the movement of amino acids against their gradient. They both move in. Sodium moves out into the blood using this transporter. Amino acid uses an amino acid transporter, moves into the blood, and off it goes. So all glucose and all amino acids are reabsorbed this way. We do not want glucose or amino acids in our urine, and if we do, that's a sign of kidney damage. So here are some notes that you can sort of write down just to sort of help you explain. I'm not going to go through them specifically, but here they are. So again, it's just using the pump, lowering the concentration so that it's higher in the PCT to then bring glucose in and then move it along into the blood. So you can pause this, read this, and make notes from this. So one thing we didn't talk about was water. So I've added some water molecules here. So the movement of all these ions into here will obviously decrease the water potential of the cell because you've got, lots of glucose and sodium constantly moving in, and also amino acids as we looked at before, and probably some other ions. Because of that, we're going to create a gradient for the reabsorption of water by osmosis from the lumen of the PCT into the blood. And there's a golden rule that you need to always remember with water movement, and it's that water follows the movement of ions. So where there are ions, there will be water. So if the ions are moving along into the blood, water wants to move along with it, which makes sense because it's going to go from an area of higher water potential here to an area of lower water potential here, and then off into the blood for reabsorption. So the water molecules will move freely across because they're not limited by any need of a transporter. They can move freely across into the cell and then into the blood. Some of the water is reabsorbed, and obviously a lot of it will remain because we need the water obviously in our urine, and how much we reabsorb, it'll depend on the requirements of the body. So water enters the epithelial by osmosis down the water potential gradient as the ions lower the water potential of the cell. Therefore, the water can then move into the epithelial cells, and then that will increase the potential in the cells, and then it can move into the capillaries. And we also reabsorb some urea and proteins. So we reabsorb half of the urea. It's important we only get half because we need to reabsorb the other half much later on. And then small proteins can be reabsorbed at the base of the epithelial